Yeah.
Actually,
Yeah,
Jan
just a second.
and Jen, if I can interrupt, on
Sure,
the screen
sure.
now to the left-hand side is an Umbellula sea pen. Oh, I can draw it over there.
Yeah.
Mm-hmm.
Yesterday we tried to sample one, but when the box came up, there was nothing in there. It might have floated out on the seafloor. Do we want to grab another Umbellula?
Which one is it? This thing?
So
Yeah.
it's this right here, where the lasers are right now.
Yeah.
I figured
What do you think, Jan?
we'd probably see another one, so this is an opportunity here.
We're kind of out in front, so this would be good if you're going to stop.
And the ship's currently holding position.
And I don't think the Umbellula is needed for this particular area.
Okay.
I think it's a mistaken sample, the coast-wide sample plan.
Okay.
Copy that.
Great, thanks.
On the move again.
So no sample?
Thank you.
No sample.
Copy.
Yeah, I wasn't on watch, but my understanding yesterday was they had collected an Umbellula sea pen. It was very small, maybe less than 10 centimeters tall or so, and they had put it in the starboard box. And we looked last night, and there was nothing in that compartment afterwards, so my perception was that it probably ended up floating out.
Yeah.
Small things tend to do that.
Confirm, Sam, the heading is still 090?
090.
Cool.
Still way out in front, though.
Yeah. Kind of looking for something to take a peek at.
Quick question for everyone in the van. At any point, have you heard Jan and my voices
Need to
echoing?
lateral over that way.
Yeah.
It's been pretty good. Yeah, it's been pretty
Every
good.
now and then I've heard it, but it's been very faint. I don't hear it at all now, but I guess Chad saw a tweet that someone who was watching was finding it distracting and heard it earlier, but none of us are hearing it now, and I was just curious if you've been hearing it.
Right now,
I do hear it every now and then, and I thought it was coming from Jen's computer.
From my computer?
But I don't hear it now. Yeah,
I haven't changed
but I don't hear it
anything.
now.
Okay. And I had checked a while back when I was hearing it, and I don't have any audio playing at all, so not sure.
Yeah, it might be echoing back to us through something in the van, maybe.
At one point, when I watched the Olympic Coast and I was on quad view, the audio was echoing, but it wasn't doing that on any other channel. So I wonder if it could be on the viewer side somehow. But
Yeah,
it
and
sounds
Guy
good
was
to me right now.
noting it earlier today too. First thing in this morning when we got on, when we both got on this shift.
All right, let's zoom in on this guy.
It's an interesting
looks like
posture.
it's going to take off any second.
Yeah.
Settling down.
If the sediments were even looser, that kind of body movement would allow it to start to hide.
So this is the Dover sole, correct?
I think so.
Yeah.
I'm looking at the eyes and the fin shape.
All right, good captures.
Cool. All right, let's come wide.
Tom confirms yes, Dover sole. They've been one of the flatfishes that we've been seeing continuously throughout the Dover and the deep sea sole. The deep sea has that more darker
Just send it
speckling.
back up
Some people
again.
say it looks like a carpet, Persian carpet. And if you notice, the body is a little wider
Thank you.
and more rounded, and the Dover is a little bit longer and narrower in shape.
White shell.
What's our depth?
820 meters.
Thank you.
Good morning, this is Justin. Am I coming through?
Good morning,
Yep.
Justin. Loud and clear.
Good morning, Justin. J-ho!
Morning, everyone. Happy World Octopus Day. I enjoyed seeing that.
Your cohort, Amity, would gave us that heads up.
I feel like this is something that I should have known before. I'm just coming late to the party. Everyone knows it's World Octopus Day.
It's is part of a longer cephalopod awareness days. So,
So
I
there's a good
think
example of a deep sea sole, and then to the upper left we
What?
have a Dover sole.
Cephalopods
Kind of
Day?
compare how they look.
cephalopod awareness days. So there's Nautilus Day, there's Squid Day, there's
wow.
Cuttlefish Day. I know.
The same week,
But-
or?
Yeah, it starts October 8th because of octopus.
Haha.
Ah,
I get it
Oct
now.
and 8.
Roger.
I think Nautilus might be 9. I can't
Oh.
remember. The internet did this.
Yeah.
It's again one of
So
one
Jen,
of
would
several
this be
cephalopod
Someone to start a
celebrations.
transect, maybe? If we keep seeing mostly soft like this?
Do we want to do a transect? Is that what I heard?
Jen,
Al, let's
did you
zoom
want to
in.
do a transect? It
Jan?
looks good on HiPAC.
Okay. Do we want to do a transect along the contour line then? From
Yeah.
where we currently are along this line?
Yeah, I think so.
Perfect.
Look at it looks like little worms on the legs of that crab.
Wow.
That's a nice,
Oh.
like, it's like a crab tattoo almost.
Tube worms. Calcium carbonate
All right,
tube.
let's come wide again. Sorry.
It's neat.
Are those
So
like a
I'm
parasite, or are they just?
not hearing
Repeat
Jan.
What the new heading was?
They're
Sam?
just
But it would be
serpulid
great for
worm
us to try to get a
tubes.
soft bottom transect. We don't have any of those yet for this dive, Pioneer Canyon dive.
Sam, can you hear me?
Yes, I can hear you.
Okay, so
Sam, what's our
Jen's not
distance to the
hearing
next
me.
waypoint?
Yes, I think that's correct. I think
Or temporary waypoint, I should say.
Jen and Jan might not be able to hear each other right now.
Can anyone hear me?
I can hear that.
I
Yeah.
don't see you.
Yeah, we can hear you.
Say 3C, I don't know what that is, but they just locked back in 3.5 minutes ago.
Samantha, can you hear me?
I can hear you, Jan.
Okay. I am not hearing the van, or so I am going to come right back.
Copy that.
She can't hear you.
Oh, they should, yeah.
So we're just holding here?
Yeah, let's hold here until we can confirm what we want to do next. If there's anything.
Yeah. So, Sam,
Yep.
let's go ahead and start what appears to be and going to continue to be a a soft transect along this contour.
Copy that. 120
So
meters.
not- Yes, please.
Alrighty. Pilot, so it'll be 108 for our heading.
108.
Bridge nav.
Oh.
40 meters, 108, please.
Oh, that's.
Is this transect 7?
That look like 108?
This is going to be number 8.
8. 8.
Yeah, close enough.
And we'll say it's in soft.
I think we might have spoke too soon.
Yeah.
Those rocks, they hear you say soft bottom and they just move right in.
They pop right back up. Okay, let's go. Let's just abort this soft transect.
Copy that, abort transect.
And is that line on the seafloor again? Going out to the left? Lower left?
Yeah.
Yeah, between those rocks there looks to be some line.
What do we have up in front?
Okay, so transect has been canceled. Do we want to continue along this contour?
No, let's continue our plan to go up to that waypoint, that temporary waypoint.
We have, we're currently at the waypoint that we had set, so I can set a new temporary waypoint along this contour line.
Okay, I'm sorry. I have to get out of my high pack because it's not refreshing.
No worries.
There we are. Yeah, let's continue to the east along this contour.
Copy that.
And Sam, when we get down to that dark pixel,
Sure.
let's go, let's try
More
and go
fishing line.
up a contour.
Okay, I just dropped a temporary waypoint there.
Yeah, I can see it now.
Perfect.
That might be good.
Looks bio-y.
Pilot, same heading for now.
Of 008. I mean 108.
108, correct.
We do have the slightest of echoes. Still, I think it's from the shore side.
Do you hear it when I'm talking? Or do you hear it, is it like a delay?
Falling off the side there.
Yeah.
It's a delay. Because I don't hear it. And when somebody else is talking, like when Jen's talking, I can hear a delay. Or when Bob, is it Bob? I think it's when somebody on the front row is talking, I can hear it. And Justin went off. And I think Jen is off, and she's now unable to hear SPL. She's going to reboot Jen's going to reboot her computer. So Justin, if you're listening, go ahead and join us while this takes a while. This is going to take her a while.
Thanks for that, pilot. You read my mind. I was going to ask for that.
Yeah, so me too.
Hey, Jan. I'm on. Can you hear me?
Yes, yes. We can hear you, Justin. And just double
Got
checking
any more zoom on
with
that?
the, it's a pretty combo.
No, other way.
Yeah. Will you zoom back out? Oh,
You want me to get it? You drive?
lateraling over.
Yeah, why don't you plan it? Gotta be quick though.
All right, let's zoom.
So are you able to turn the camera side by side, the Herc camera? Or is it, oh yeah, okay. Pan, what is that called? Pan versus tilt.
All right, let's zoom out. Gotta get back ahead.
Gotta get going again.
That's a pretty picture though.
Yeah, we got some good
With the thorny
captures.
head next to it. We can go a little bit lower to get the tail of the fish. It's off my screen. There we go. So Justin, have you seen any of the new information about the what is it, was it the Iron Crown pottery?
Ironstone, is that right?
Ironstone.
Yeah, I've been following along. Chad's been keeping me in that loop, and pretty interesting. Logo matching, shards of when people think about 1800s pottery, guess where their mind goes? Yeah, I got to be part of that. That was pretty exciting.
He
And
also
I saw that
sent
Bob
around.
Schwemmer.
Yeah. What did Bob have to say? I did not get to read Bob's emails.
I think he mostly was kind of interested to see it and to see the sleuthing that some of our online watchers and
That's
the
kelp.
chat.
Kelp.
Yeah, there we
There's a
go.
bunch of kelp.
Do we want to stop here?
Yeah.
Bridge nav?
Got my eye on
Am
you,
I
Kelp.
close enough? Or should I get a little bit closer?
Oh, look at that. Macrocystis.
This one's not good away, I'm pretty sure.
All right, I think I'm done. Will you hit the stick lock?
All right.
Is that good?
Yep.
All right.
Did Taylor Ann already present her microplastics live event?
Look at
She
the
did.
lasers on it too.
Yeah, it was last week.
Oh, is that last week?
So what's the plan here for this?
You want to maybe try and do like a cut and slurp type of
Yeah,
thing?
cut and slurp. Yeah, just a leaf instead of a whole long
Yeah.
bit of it.
State.
What jar are we gonna go in?
Whichever one you want. The slurp has some in it already, or the flush has some in it already, so any jar is open.
Okay. Get the cameras here. Bucket.
So we want to swap the sample jars, or is this?
Yeah, we want to get off of flush. And go ahead and run it, and just to get the jar. Yeah, just pick a jar, any jar, and then just run it.
Oh yeah. And that's the starboard suction, right? Just up to.
So that's number 6, I think, right?
Oh.
Yeah, T6.
I'm back. Can you hear me?
Loud and clear.
Yay. Hi.
We can hear you. Can you hear us?
Woohoo. Yay.
Alright.
Yes.
Sound great.
Thank you. All right, great.
You can
Now,
cut
is
the
the van
beans.
still here?
That looks good.
All right.
I've been following along and I thought, now here's some kelp that won't get away, right?
Gotta watch those kelps. Never know.
If this is able to run away from us, we've got problems.
Yeah.
Since it was a big specimen, I was waiting for the 8-inch slurper. I remember it was like 1.5 inch the last time we tried this, and I was thinking this, but I don't think that exists. We don't have an 8-inch slurper, do we?
Not yet. Maybe a funnel attachment.
You're just gonna bring it right up to the slurp?
Yep.
Should I hit it? Or not yet?
You can hit it.
It's like a lot of growth on that macrosynthesis.
We yeah. How many beans you got? You got full beans?
We're at full beans.
Get off of there.
They're wily. Use kelp.
They are.
I think it's hydroid growth all over it.
Yeah, this one's been here a while.
Yeah. Is that enough of it, or you want more?
There's something in there.
There
There
it is in the,
Number 1.
Yeah, kill it. Let it
yeah.
settle out. You want more, or what are we thinking?
Yeah, maybe go for a little bit more.
A different sample jar or same one?
Same one.
Okay.
Whatever you can get.
Yeah, that way.
So Steve, when they get done with this, I'd like to hear about what Taylor Ann is planning to do with these samples.
Sure.
I don't want that piece. Fish is taking a lot of interest in it. Rock.
Nav, just for your reference, this is 21021.
You are reading my mind. And you include cruise ID and
Yeah.
dive number?
Yeah, 122.
Great, thanks.
You don't need to put the dive number.
Okay.
Ready for suction?
Ready.
I think that's it.
You think it's in there? Oh yeah,
I think so.
I think now it is. Does it look like there's 2 separate chunks in that jar?
I think so.
Yeah, I think so.
Okay.
Is that enough?
Yeah, I think that's enough.
All right.
Get back up to the.
Yeah. So hit stick lock.
Yep.
Yeah.
And then don't really give it.
Get up before I go forward.
Yeah. Just
Yeah.
kind of let it drift up, and then you can go.
Is that a sable fish right there?
Sablefish.
Hmm.
Let's zoom in a little bit, Al.
Four 0 meters, 108.
Oh, a little bit out.
108.
So yeah, we've
Alright.
been collecting some various different types of water and specimens for microplastic sampling. Taylor N has been doing some water sampling
let's
from
go
the
wide.
Niskins, basically trying to filter out microplastics from the water column at various depths, which we accomplished last night. So she's going to be taking that material back to the lab and examining it for microplastic concentrations. As well, there are a few specific items she had on her list to collect for microplastics examination, namely kelp was one of them. So it's thought that the mucus and blades of the kelp can attach, we can get microplastics attached and trained on those plants, and then when they fall to the seafloor, they carry that microplastic with them. So she's doing that both in shallow water and now in the deep sea, so she has a reference point to compare to. And we've also been on the lookout for both sea pens and sea pigs for microplastics. There's some consideration that these animals might be collecting and consuming microplastics from the water column as they fall onto the seafloor. So she's going to be on the lookout for those over the course of this cruise leg and the following one down in Channel Islands as well. So those are just a few methods, but she's has some very detailed analyses also she's doing at the surface, where she's taking surface water samples every dive site we're at and collecting and filtering water samples from there, so we can compare the concentration of microplastics across the depth in the canyons.
Same coral or sponges.
Yeah, I'm hoping to get some of those samples of the microplastics when we're at Davidson as well. We have, last year, we were able to filter water and get sediments to look at persistent organic pollutants. But we don't have any information about microplastics out at Davidson, either in the water column or in the sediments or in the benthic animals. So that would really help to fill a gap in understanding for the management zone around Davidson Seamount.
The line or grass there on the left?
These remind me of seagrass, but you're right, they're kind of long. So
I guess that's not line, that's just some sort of algae of some sort.
So Sam, while I was having technical issues, I wasn't able to hear what y'all were talking about. I saw in the chat that there was an intention to do a transect, but it seems we're not on transect. Could you catch me up?
We had originally started a transect that was requested maybe 20 minutes ago. And it was decided that it was not the soft sediment that we were looking for, so it was canceled.
Got it. Okay, thank you.
So we're actually continuing pretty much to the same location that we were planning for, just not as a transect, since we have quite a bit more rocky sediment than anticipated.
Yeah, okay. And Chad agrees, he thinks that was also, so Phylospadex is the scientific name for seagrass.
Which is the one
That's what it looks
true
like
plant in the ocean, is it not? As an angiosperm?
Yes, it's an angiosperm, that's right. So there's sea plant, but that's the only one. Well, there's a so there sea grasses is kind of the general term, and then we find them in estuaries and quiet waters, shallow waters, and we also find them on the along more of the coast and at the deepest part of the rocky shoreline, and we commonly refer to that as surf grass. But they all look the same, these long, thin blades. And they provide great habitat in the shallow waters, all sorts of, particularly like juvenile fish and crabs.
What are we doing? What's the heading?
108. Same heading.
Looks like we got a couple sponges on the left, bottom left portion of the screen. And a thorny head peeking out from under an overhang.
Jen, I was going to ask how many sponges are you seeing on this cruise? As many as you expected, or I know that we were talking a little earlier about not seeing too many this morning.
I barely saw any yesterday. I mean, there may have been like really small ones encrusting the rocks, but as far as the larger sponges, given how many corals we saw yesterday, I was surprised to not see more sponges. And we're seeing a few this morning, but not a ton. Oh, there's a skate upper left. So I guess in this habitat, that's more
this current.
right now. So this looks like a rough tail skate.
Yeah, I've been watching whenever I can, and it seems like I've been only seeing rough tails. Have you seen any other species of skates?
Let me think. Mostly rough tails. I'm thinking, I'm trying to remember not say what I saw when they were up in Olympic Coast, but trying to remember what we've only seen here. I think I've only seen the rough tail skate so far.
Cool.
Of course, I haven't been watching. I've been watching the entire time during the dives. I have chosen to sleep some. It's hard to sleep. I mean, good stuff's found while you're sleeping and you miss it. So
that is true.
There is a bit of fear of missing out associated with 22-hour dives that I've been wrestling with myself. I think I got 13 or 14 hours of day one, but you got to pace yourself a little bit.
That's right. FOMO is real.
Yep. FOMOD, fear of missing out on dives. I guess then it's FOMOD.
Tom's chiming in saying at these depths, rough tails would be the most common to see. Thanks, Tom.
Hopefully people noticed when we zoomed in that you could see the line of spines going down the tail of the skate. And that's one of the things we're looking at to make that identification.
I know one of the goals for this Pioneer Canyon exploration was to kind of look into animal interactions and associations. That's kind of why I was asking about your wild speculation on why we might be seeing lots of coral diversity and fewer sponges. I don't know if you have any other thoughts on that, qualifying it as wild speculation.
No, I'm used to,
let's come back
given the previous dives I've been a part of, seeing a lot of sponges, actually more sponges than corals, and particularly when we see corals, seeing lots of sponges around as well. So that's why I found that interesting. Now we aren't seeing that many sponges here. We're seeing some, not a lot of corals or sponges, but we are seeing a little bit of each. That's more typical of what I've seen in the past. But I was trained as more of a near shore, an estuarine ecologist. So these past few years, when I've been involved with the Nautilus cruises and I've gotten to go out a little bit with Mbari and on a NOAA ship, the Shimada, I've been learning so much more and getting to see so much more of the deep sea. And I really love it, and just trying to continually learn more and more. about how to identify the animals, what lives where, how these animals are associated with the habitat and with each other. I think a lot of people have the preconception that there's not a lot in the deep sea, and particularly in areas with soft bottom, but as we can see on these dives, the soft bottom is teeming with life as well.
So science, we are just completing that move to the temporary waypoint here. Do we want to stop here and take a look around or continue along the contour?
So let me just check in. Jan stepped away for a moment a little while back, and let me see if she's back. And if she's not, then it's if I don't hear back from her in a moment, I would think we could continue in this direction a little bit further, and then ahead we have an area that looks to be a bit steeper that we could go up.
Up towards the mystery zone here doesn't have a lot of good data.
Well, it'd be interesting actually to hear from Guy if he's interested in going through the mystery zone to get more information on that, or we could repeat. Okay, I'm getting a message that Jan is going to come back in a moment.
All right, Al, let's zoom in.
Almost look like a line from the distance, but just really thin rock. The little nudibranch there, bottom, just under the lasers now.
Yeah.
All right, let's come back wide.
Is that the same?
So continue on the line for a little bit while we get our next steps in place.
Okay. There's no particular interest in the mystery zone.
Yeah, can we get...
Oh, hi, Jan.
Hi, I'm back. I'm sorry. Yeah, close-up of this nudibranch. I don't think we've seen this particular one. Oh, we're moving.
Oh,
never mind.
What?
Should I go back? Can we go back?
Yeah.
Sure, if you can.
Let's find out if I can.
I had
Ship's
what I
away.
thought was going to be a quick chat with our analyst, habitat analyst with National Marine Fisheries Service,
So it's right over
and I think this is a good area to start collecting some more transects.
Left side there, left of the
there. Yeah,
inhabiting.
I'm full.
Just turn your
Lateral.
head that way.
Yeah.
Jan, is this soft enough to qualify soft for
Yeah, yeah. We
Okay.
were talking about kind of balancing between the more standardized soft mixed hard versus when you get into this area of pretty kind of uniform and still wanting to characterize the entire area. So we're going to go ahead and collect and just see what we can see. If the substrate is kind of uniform along the entire 120 meter transect, we can end the transect. And if it kind of gets kind of jumbled up between mixed and harder or mixed and a lot of mud, we'll go and kind of extend it to about 150.
Is that close enough? Or not quite.
Are we looking? What are we
And
looking at?
I'm not
little
back in chat yet,
Oh, the
so
nudibranch.
I'm going to get back into chat.
Trying my best.
Are we trying to collect this nudibranch?
No, I just would like some close-ups of it.
right.
And Jen, I'm not monitoring chat right now, so
let's
it's,
zoom in
yeah,
out.
I don't think we have a
There?
request.
You happy?
No, we have not received a
there you
request
go.
for it.
Okay, if we could just get some close-ups of it. I don't think we've seen this one. We've seen the larger
Oh.
Drifted off.
coral,
Yeah.
coral-eating ones.
Down. Ah!
Oh,
Blew that.
have to look for another one of those nudibranchs. Okay.
Too many cooks in the kitchen on that one. Sorry.
All right. So we will be keep an eye out for those nudibranchs. So I hear that transects might be of interest.
Yes.
I need a lateral over.
Yeah.
Okay.
Current's
So
pretty stiff.
along this contour line to the east, or bumping up towards the mystery zone?
Yeah, I'll turn around and go
Let
forward.
me check high pack here. Hold on.
Yeah, currently we're on a waypoint, a temporary waypoint that starts with a 1713, and it's where the ship and all the other icons for Hurricane Argus are.
Got it. Okay. And what did Guy say about the kind of mystery spot where we have some missing data? Like I think I would like to move over that area
He said
just
no
to see what's
specific
there.
interest in the chat a few minutes ago.
Okay. Okay.
What's the heading?
Heading's 108 for now.
Still 08.
Scratch that, Bob. Let's make it 090 while we settle out.
0908.
But ship's stationary.
So yeah, if we could. So Sam, if we can go along this contour, I think that'll be fine. We don't have to follow it exactly.
Okay, great.
What I really would like to do is get to that those tight contours that are a little bit further to the east, kind of almost directly east of the ship icon, and then go straight up.
Okay, I just dropped a temporary waypoint there in between the two narrow contours that I think you're talking about. Should see it in a second. Or seconds.
Let me close out my high pack. Yeah, let me close out my high pack and see if I can get it to refresh.
I see it, Sam. She may want to go even a smidge further, but let's wait till she.
Yeah.
In the meantime, Sam,
there it is. Yeah.
What's the distance?
Yeah, I think it's
just a fish.
I think that's a good.
Just a fish. It's just an old fish.
Yeah, I think that's a good waypoint.
Okay.
Temporary waypoint.
And that's 200.
Because we are getting closer. We are getting closer to 2S, right?
Correct. It's up in the top right corner at the moment.
Great.
So this next waypoint is 245 meters. Alrighty. Bridge nav. meters, 094.
What's that? Coral.
Pilots were doing 094.
Roger.
All right, Al, let's zoom in on that.
Looks to be another Isidella.
Oh, nice.
All right, let's come wide again.
And Steve, this will be transect 8. Is that right?
That's my calculation also.
Is that another gastropod egg tower that we see coming along the yellow structure?
Ooh, yeah.
Neptunia, was it?
I think so. Yeah, the Welk.
Yep.
For the yellow eggs, yeah.
We did.
This is a
Alright,
120 meter
let's zoom in.
transect.
That's correct. If we run into a lot of changing substrate,
And
we'll go
push
to
in a little bit more.
150.
Copy that.
Nice. Good captures on the egg case.
What's
Alright,
on
let's
the
zoom
rock
out.
surrounding it?
Oh.
Is that sponge or a little Nidarians? I wonder.
That little orange one.
No, it's just
Oh.
the kind of frilly grayish white that's kind of going all along the edge of that rock and is on the surface of the rock below to the right.
Yeah, let's
So we
zoom
see it
in.
on a number
it-
of these rocks,
is it
actually.
debris?
You see?
Say that again, Jen.
Well, I'm just trying to see what it is that seems to be lining a lot of the right sides of these rocks. You see that? Just trying to
Yeah, I
determine
see
what
that.
that's sponge or some sort of Nidarian or
Need to
something else.
get planted.
Yeah.
Looks to me
Come-
like it's
come
some
light
sort
off.
of sponge, maybe encrusting sponge.
Yeah, maybe. Interesting. Just hadn't seen that in some other locations. But if you look at the rocks in this whole little area, you see that the right side appears to have that.
Yeah, there's a significant amount of it, Jen. I appreciated your comment about that. It's kind of all over this area.
Just right there. Kind of interesting. Reminds me
I
of
am
how when we
planted.
found the octopus gardens,
Don't
there
let
were
off
certain
on the
encrusting.
forward. You want to be.
Yeah.
You want to just
Do
keep
we want
everything
to
steady state so that it's real
Stop to be able
stable,
to take a closer look at
and
this?
then try and reach over with your finger,
Potentially a sample?
hit the stick lock.
I'm not sure if that's anything that anyone in particular was interested in. It was
Okay.
more just catching my eye as a
Yeah.
pattern.
Oh, also technically we were on a transect.
Oh.
Oh,
Oops.
oh, I didn't know it
Oh, we
started.
had
yet.
started?
I didn't know.
Sorry,
I didn't know it started yet.
let's just
Sorry.
no, it's okay. Let's just restart. We can start here if you'd like.
Okay.
Since we had that surprise. I think we all got
I didn't see anything that Steve noted.
Yeah, my impression was we were going to start the transect when the ship was definitely moving and the vehicles were moving rather than stationary. So that's what I was waiting for, that cue.
Okay.
That was my understanding also.
My bad on that one. We will start here.
All right, so
Alrighty. Bridge nav. (30 meters 094). Okay, transect is starting. 120 meters to the east.
Interesting looking stuff on the rock that's passing along the right side as well.
yeah, I think those were clusters of gastropods. I saw a bunch of snail shells. That makes me think that those features you were looking at before on the right side of the rocks there, or maybe egg cases also.
Oh.
They did look really familiar. I think we've seen them before. You're right about egg cases.
Yeah.
Some sort of snail.
Yeah, I might even lean towards egg cases at that point rather than sponge.
Yeah.
Mm-hmm.
I think we have actually seen those egg cases in spiral formations on rocks. or a similar species. I think we even have a highlight video of it, but I'm not able to access in my brain where that was or what kind of species.
Bias that way.
Yeah.
I think we might have seen it in Olympic Coast or potentially Greater Fairlands, actually.
Well, it's interesting how many gastropods or snails that we've been seeing in these two Pioneer Canyon dives. It's really remarkable, much more than I've seen elsewhere that in dives I've been associated with.
Jen, you were talking about how fascinating the deep sea is for you. For people that are just joining in and maybe this is their first peek under, this might be their first time seeing a thorny head or their first time seeing these mushroom corals that we've been passing. Those are mushroom corals, aren't they, that we've been transecting
We have seen
past?
some mushroom corals. Definitely, they look like kind of like a golf ball in that they're round and then appear to have kind of pock marks or maybe even a wiffle ball, if you're familiar with a wiffle ball. But then some of them have the polyps out, and so you see those extending from the surface.
Yeah, I think I got to see maybe 3 or 4 at the beginning of this transect on the edges, on the margins. So that might be something people can look out for.
But see here, look at all these snail shells. It's just amazing. That's not, you know, typical for parts of the deeper seafloor that I've seen, but I have to say that most of the cruises I've been involved with are deeper than this. So it's fun to be at this depth of 815 meters because through the deep sea, certain species are found in certain depth bands. And so out at Davidson Seamount, we're looking at depths that are much greater than this. So it's nice to get a chance to be here in the, yesterday we got down to say 1,200 meters. Now we're up at 800 meters and see what lives in this depth along the seafloor.
Uh-oh. Missing Jen.
We're getting some input on. What's that, Jan?
You're cutting out on my end.
Oh, do I sound all right in the van?
Loud and clear in the van.
Sound
Okay, thank you.
marvelous.
Yeah, I think it's.
And
it's
you
just
look
my bandwidth
marvelous.
here.
I'm getting that echo again. Just started up.
We're not hearing it here.
So a couple things came to us in the science chat. We have both Tom Leidig and Kurt Whitmire agreeing that those could have been whelk eggs on the rocks. And then Tom saw a little poacher go by, so I'm going to keep my eyes out. Poachers are adorable little fish. And they might be hard to
How
see
small are they?
in a transect. But we can keep our eye out once the transect is over, see if we find any. They look a little prehistoric to me, which I think is really neat.
What's our altitude?
So we're at about 2 meters right now, altitude.
Okay.
Going between 1.8 to 2 meters.
I'll try to
Okay,
come down a little bit.
if we can come down a little bit, it's hard to see out in front.
Yeah.
This high up.
And Tom has commented that poachers are less than 20 centimeters long. Looks like we have a skate that's going to go through the right side of your screen. I'm going to assume another rough-tail skate at this point, given what we've been seeing and what Tom let us know about it being the most common of the skates at this depth.
And Jen, Tom shared with me that other skates we might might see would be a Pacific white skate. They're deep, but they can get as shallow as 800 meters, and a deep sea skate as shallow as 400 meters. But many of these shallower skates have depth ranges that extend this deep, but are rarely seen this deep. Thanks for that, Tom.
Okay. But as Justin was saying, most of the fish we're seeing right now are either flat fish, right? The ones that are laying close to the seafloor, look like they've been, compressed by someone pushing down on them. And they're pretty good at blending in with the bottom. These are Dover sole that we're passing over now. And then we're seeing a lot of thorny heads. That's ooh, look at that thorny head's putting up its fin. And to my eye, it looks like it has a longer third spine. So I think that would be the long spine thorny head.
So Jen, you were saying earlier that you don't dive this shallow, or you're not accustomed to seeing habitat this shallow because your dives are typically deeper. Are those on Bari cruises?
Well, I've had the pleasure of going out with Bari, the Monterey Bay Aquarium Research Institute, once, and we went both to Sur Ridge, which is this kind of rocky ridge feature south of Monterey Bay. And then most of the diving that I've gotten to do is out at Davidson Seamount with the Nautilus. But I also, on that Bari cruise, we went to both Sur Ridge and to Davidson Seamount. And I haven't been on any of the cruises where we were able to look at the top of the seamount. We were going to do that one year with the Nautilus, but due to weather and equipment errors, we weren't able to do those dives. So I've mostly been looking around the base of the seamount, which is very deep, 2 miles deep. So we don't see all these diversity of fish down there. So it's fun to get to see a wider variety of fishes at this depth.
Yeah, see, now I'm used to diving much shallower. So Cordell Bank, Rittenberg Bank, some of the rocky reefs like Cochran Bank, even Farallon Escarpment is shallower than this. And so I think this is, oh, wow, this is really, really deep. But I'm really excited to see Davidson because I haven't been on any cruises at Davidson. So it'll be quite an adventure for me.
Yes, and all these depths that we're looking at are below where sunlight has an impact. So it's perpetually nighttime, at least from the standpoint of light. And these animals are used to being in the dark all the time. So it's all really deep from the human perspective.
Speaking of being in the dark, for those of us who love field guides, people might not have a field guide to the deep sea or to Pioneer Canyon on their library shelf at home. And I just wanted to mention that the Sur Ridge Field Guide, which has come up on many shifts by many people, just want to mention it again. It's put out by the Office of National Marine Sanctuaries. It's called the Sur, S-U-R, Ridge Field Guide, and you can find it online. And Monterey Bay National Marine Sanctuary put it out. And it is a real wealth of information. A lot of the people who've been commenting or on the cruise have been referencing it or looking at it. So, and one of the authors is Erica Burton. I just wanted to mention that for everybody. Check it out, Sur Ridge Field Guide.
That's a great tip. I'll make sure to add that to the Nautilus Live website.
And while you're finding that, Sam, you can, because the Sur Ridge Field Guide has a lot of the animals at the depths that we're kind of looking at now. And then when we get out to Davidson Seamount, there's also a similar document for Davidson Seamount, and that'll include those animals that go even deeper. So the depths we find out there at the seamount. Mm-hmm.